To the lecture online and whatever it happens? Yes, sounds wonderful. I like to think there's actually some value added to me being here and you being here. Again, I like to think that.
You're adding to each lecture a little each year, right?
Sometimes a little, sometimes a lot. Yeah. What am I adding to each lecture each year? Which unfortunately means that might actually be on an exam. Yuck. So, plant viruses. This particular plant virus here is CCMV, cowpea chlorotic mottle virus that we'll talk about a little bit more. I really like this virus for a couple of reasons. One is, of course, it's this beautiful T equals 3 quasi-equivalent icosahedral structure, which everyone's really happy about. But more importantly is that it has this really fascinating conformational change that takes place. Basically, sort of expands. I don't know how many of you have seen these kids' toys with the wonderful expansion, contraction kind of thing. It's exactly what this virus does. It's so cool. So we'll talk a little bit more about that later on. The professor who I worked with during my postdoc worked on this and has done some really cool nanotechnology kinds of stuff with this. So yeah, we'll talk about RNA viruses. Again, plant viruses, totally not doing them justice in this period of time. The bromoviruses or the bromosaic viruses, the cucumber mosaic viruses, particularly that's what the chapter is about and one of the ones that we know the most about. We mentioned already that these guys have segmented genomes, but unlike the segmented genomes in flu, which are all in one virion, they're split up into multiple virions, which is just crazy that this is how evolution has worked for these things. One of the really critical things about these plant viruses, and probably why they can evolve to have these segmented genomes, is it's actually really hard to infect a plant. If you think about a plant, it's sitting in one place for most of its life, unless you have distribution of seeds and that kind of thing. So it's evolved to be really good at protecting itself, not surprisingly, because it can't run away. So once you have a virus infection gets into part of a plant, getting to other parts of the plant is really kind of tough. And so all plant viruses have these so-called movement proteins where they can get from one part of the plant to the other. So this is really quite different compared to any of the other viruses really we'll be talking about right now. We're all mushy and easy to get around in, but plants are definitely not that case. And then at the end we'll talk a little bit about TMV, and again, a little bit because this is the book about TMV, actually one of the books, and only from 1930 to 1965, so 35 years of TMV. Why is TMV so exciting, among other reasons? First virus ever discovered. So, and that was well before 1930. So again, our key concepts, most of the plant viruses, interestingly enough, are single-stranded RNA viruses, positive sense. Very few negative strand, very few double-stranded, few single-stranded DNA, very few double-stranded DNA viruses. So quite why that is still kind of an open question. Almost all of them have segmented genomes. Again, not all of them TMVs, and of course an exception to that. Talk a little bit about plants and why plants are different, mostly again because they can't run away. Conformational changes in the virion. We looked at that already with CCMV. We'll talk about it a little bit more. What happens to the host on these virus infections? And then I wanted to tell a really cool story that actually is not really a plant virus. It's a fungal virus, but it has some really fascinating effects on what happens to the plant. And then we'll talk at the end about some helical structures, particularly for TMV. We sort of skimmed over them really quickly when we talked about symmetry at the very beginning of the course. So the title of the chapter is Cucumber Mosaic Virus. We'll talk about that, but we'll also, again, talk about a number of these other ones. Where did it come from? Again, this is mostly the viral disease. The structure, which is this beautiful, again, T equals 3 quasi-symmetric structure. How does it get in? This is probably one of these things that I probably should have highlighted in red because it's clearly very different relative to all the other viruses we've talked about so far. Replication is actually not too terribly different than a lot of the other RNA viruses we've talked about, single-strand RNA viruses, and packaging. Some of the big differences, again, we put it in the second column over here, are satellites. Satellite RNAs, they're not really satellite viruses because they don't have any virions. They're just these RNAs that are floating around together with the virus. They need a virus in order to be able to replicate to cause disease, and they can modulate that disease. They don't even code for usually any open reading frames. And so how this little RNA could have so much influence is really pretty impressive. Talk a little bit more about CCMV and some of the applications here of that conformational change, some hot viruses, because hot viruses are cool, and tobacco mosaic virus. So this is a reminder. We're talking about positive strand, single-stranded RNA viruses, which of course can also serve as messenger RNAs. So all you need to do is get that RNA inside the cell. Once that RNA is inside the cell, as long as it looks okay, and what looks okay in terms of a messenger RNA, what do you need? Caps, and to some extent tails, but most importantly caps to get your translation, this little double blob here, the ribosome, for going on here. So these are single-stranded RNA viruses. The vast majority of viruses that people have studied in plants are these single-stranded RNA viruses. What does it look like? This is a cucumber that's been infected by cucumber mosaic virus. A mosaic phenotype just basically means that you've got bits which are green and bits which are not green. If you go back and look at Biojink's original paper in 1898, if I remember correctly, he talks about albinism. And that's because lacking the coloration here. So this is a really classic kind of phenotype for your disease. Cucumber mosaic virus was originally discovered in 1916, and this is sort of the naming that all the plant people use for their viruses. So what's the host? Cucumber. What's the phenotype? Mosaic. So cucumoviruses. Again, I like their picoRNA viruses, they picornaviruses. Potty viruses, so be a potato yellow virus. So that's where they come up with all these names. And I didn't bring the virus taxonomy book, but it's just filled with all of these things. And as I mentioned, CCMV, cowpea chlorotic mottle virus. So it tells you what it infects and what kinds of phenotype you actually get. Cucumber mosaic virus is really interesting because it's probably, at least as far as species are concerned, the virus with the widest host range that has ever been found. Last count, something like 1,200 different species of plants can be infected by this cucumber mosaic virus, which gets back to this naming convention. Okay, it's cucumber mosaic virus, but it can infect tobacco. Okay, wait a minute. So it's the cucumber mosaic virus infecting tobacco. So the nomenclature here, it's the first one they discovered. Yes, it was in cucumber, but infects all kinds of different things. So unfortunate, yeah, it makes sense for your first one, but if you have a wide host range, then it's not particularly good. Now, the reasoning behind the whole wide host range is still pretty up in the air. Why should you have something with such a wide host range? Because most of the viruses we've talked about for this class and in general have very specific host ranges, very few organisms, or even in some cases cell types that they can infect. Here, it's very different. And a lot of that has to do with the mechanism of transmission, how you actually get from one place to another. So again, hopefully we're not beating this too much over the head. T equals 3, quasi-equivalent icosahedral structure. Quasi-equivalence because it's one and only one coat protein, which is put together either in pentamers, so your 5-fold symmetry here, or hexamers, the 6-fold symmetry right here. So if you're going to do our T numbers, start here. H is equal to 1, change directions, K is equal to 1, that gives you your T equals 3 structure. The main part of the structure here is a beta barrel. This should look really similar to another kind of positive strand, single-stranded RNA virus with a pseudo T equals 3 structure that we've been reviewing for our exam. Right, the picoRNA viruses. So polio in particular has a very similar structure here. This beta barrel structure. Here, It's actually a little bit different relative to what's going on in polio. All of these viruses, and the cucumber mosaic virus and the close relatives, have this very arginine-rich N-terminal alpha helix. What do you think this is interacting with? So arginine is what? One of those few amino acids I expect you to remember. It's positively charged. What's it going to be interacting with? Negatively charged genome, RNA. So that's on the inside. So immediately this tells you that this is on the outside and this is on the inside of that. So how do these, again, pseudoequivalent T equals 3 icosahedral particles get inside the cell? The way they do it is getting some help. So almost all plant viruses, with few little exceptions, are vectored, i.e. they've got some other mechanism for getting inside, not even so much the organism, but even inside the cell. Most plants have these really thick cell walls. It's really tough for a virus to get through these things. Plants don't usually have all of these extracellular proteins that are going to be communicating with the inside. Plants don't do receptor-mediated endocytosis. So how do you get a virion inside a plant cell? Basically, it's by chewing through them. And this is the aphid, which is doing that chewing right here. One of the, I think, really fascinating aspect of cucumber mosaic virus and a lot of related viruses is that to get a productive infection, you need multiple genomes and multiple virions. And that's because each of the RNA segments, which is part of your genome, is packaged in a different capsid, so different virions. And this seems to be a really inefficient way of packaging your genome, because if just one virion gets in with one RNA, that's not going to be enough. A different virion with a different RNA gets in, that's not going to be enough. You've got to get all of them together. And the reason that seems to work is because, yeah, this is an electron micrograph of an aphid, but it's still ginormous relative to the number of virions which are there. You need the TEM. And so the mouth parts of this aphid, and in fact a lot of the plant-eating insects, is just absolutely covered in virion particles. You can literally scrape virions off of the mouth parts of these things and look at them in the EM. So very large inocula you're getting there. So if you're thinking about transmission, aerosols, the kinds of things we've been talking about with a lot of these human viruses, just you're only able to get a couple of virions in any of those individual aerosols. So doing this split capsid kind of thing is not going to be terribly successful. On the other hand, if you're getting into the cell through one of this real damage processes, then you can get a bunch of virus particles in at the same time. Most plant virologists don't use aphids. They actually literally just scratch the top of the leaf and then put their virus on top of it. Works just as well. So physical damage. It seems to be basically physical damage. Making a hole. Through the hole is how the virions get in. Yeah, there's a question?
Doesn't the virus have to replicate in the vector at least a little bit in order to be able to be passed on to the host?
So the question is, does the virus need to be able to replicate here in order to get passed on? The answer for most of these plant viruses seems to be no. It doesn't need to replicate, partly because many of the virions are very stable, so they can hang out for a pretty long time, and partly because usually it doesn't take very long for an aphid to get from one leaf to the next leaf, or even from one plant to the next plant.
They do in mosquitoes, right? As a vector.
Right. So